Hello students and my dear friends, myself Madhu Vancha. In this session, we'll discuss some of the important parasitology multiple choice questions part 2. First question, among the following, the sensitive method for microfilaria detection. Option A, wet mounts. Option B, thick smear. Option C, QBC method. Option D, concentration technique. So microfilaria we can see in case of Wuchereria bancrofti infection. So what is the right answer? What is the answer? That is option D, concentration technique. So when microfilaria density is low, so when we are expecting the density is low, the concentration technique such as membrane filtration technique and sedimentation technique, they help to detect the microfilaria in the blood. Here the right answer is option D, concentration technique. Next question, Elephantiasis is also known as option A, Calabar swelling, option B, Malabar leg, option C, fugitive swelling, option D, river blindness. Here the right answer is option B, Malabar leg. Elephantiasis, it is also known as Malabar leg. So if you see the Elephantiasis or Wuchereria bancrofti infection, so this disease was first recorded in India as early as 6th century BC. And that was recorded by Sushruta in his book Sushruta Samhita. And if you see in the 7th century, like AD Madhavakara, he described the signs and symptoms of this disease in his Madhava Nidana. And in the year 1709, Clarke, he called elephantoid legs in Cochin as Malabar legs. So here the right answer is option B, Malabar leg. Next question. The infective form of hookworm is option A, Redditifum larva, option B, Filariform larva, option C, Microfilaria, option D, Pyriform larva. So hookworm, that is also called as Ankylostoma duodenale. So if you see what is the right answer, that is option B, Filariform larva. So Redditifum larva, it will develop into Filariform larva. And where this Filariform larva is present, it is present in the soil and it is having the size of 500 to 700 micrometer in the diameter. And this Filariform larva, it is present in the soil. It has ability to penetrate the unbroken skin. So especially like it break the skin between the toe fingers and the dorsum of the foot and the medial aspect of the sole. So here the right answer is option B, Filariform larva. Next question, mode of transmission of Ankylostoma duodenale, that is mode of transmission of hookworm. Option A, congenital, that means by birth. Option B, sexual contact. Option C, skin penetration. Option D, inhalation. So here the right answer is option C, skin penetration. So here Filariform larva, it can enter into the host by the penetration of the skin, by the penetration of the unbroken skin. Next question, hookworm, they results in anemia due to option A, iron deficiency. Option B, secretion of anticoagulant at the site of attachment. Option C, blood sucking by parasite. Option D, all of the above. So if you see hookworm, it causes anemia. So why they causes anemia? Here the right answer is option D, all of these. So it is because of the iron deficiency, secretion of anticoagulants at the site of attachment, and blood sucking by the parasites. So if you see adult worms of the hookworm, like adult worms of the parasite causes hookworm disease. So it is characterized by microcytic hypochromic anemia. So why it leads to the anemia? Because hookworm ingests the blood and they attach themselves with the mucosa of small intestine. And adult worm sucks the more blood. And these worms frequently live from one side to the another side, which living there behind the small bleeding points. And this worms possess the anticoagulant activity, and it leads to the iron deficiency anemia of microcytic hypochromic type. Next question, which of the following is false about Ascaris egg? So which of the following is false about Ascaris egg? Option A, unfertilized egg is elongated. Option B, egg is bile stained. Option C, fertilized egg floats in saturated solution of common salt. Option D, fertilized egg has no albuminous coat. So here the right answer is option D, fertilized egg has no albuminous coat. So this is false. So if you see, these are all true about the Ascaris eggs, such as unfertilized egg is elongated and eggs are bile stained. So both fertilized eggs and unfertilized eggs are bile stained. And fertilized eggs, they float in the saturated solution of common salts. And what is a false about this egg? Fertilized egg has no albuminous coat. That is false. So fertilized egg has, it is surrounded by thick translucent shell with an outer albuminous coat in the form of the rugocytes. Next question, a gravid Ascaris female worm can lay how many eggs per a day? Option A, 50,000 eggs per a day. Option B, 1 lakh eggs per a day. Option C, 2 lakh eggs per a day. Option D, 4 lakh eggs per a day. And if you see the right answer, that is C, adult worm, it lays 2 lakhs eggs per a day. So mature female worms lays the 2 lakhs eggs per a day. So it lays two type of eggs, fertilized eggs and unfertilized eggs. So both fertilized eggs and unfertilized eggs, they are passed out in the feces of the infected person. Next question, the infective form of Trichuris trichura. Option A, unembryonated egg. Option B, embryonated egg. Option C, larva. Option D, adult worm. So which is the infective form of Trichuris? So here the right answer is option B, embryonated egg. So Trichuris trichura, it is also known as whipworm. See, this parasite, it completes its life cycle only in one host, that is in the human beings, and there is no intermediate host. So how the humans get the infection? They get the infection by ingestion of embryonated egg, which is contaminate the water or food. Next question, which cystosoma species causes central nervous system infection, CNS infection? Option A, Cystosoma mansoni. Option B, Cystosoma hematobium. Option C, Cystosoma intercalatum. Option D, Cystosoma japonicum. Here the right answer is option D, Cystosoma japonicum. So Cystosoma japonicum, it causes central nervous system infection, such as it causes neurocystosomasiasis. Next question, the mode of transmission of Echinococcus granulosis infection in man is by option A, ingestion of contaminated food with dog species. Option B, by inhalation. Option C, autoinfection. Option D, none of the above. So here, if you see Echinococcus granulosis, it causes Hydatid disease in the humans. It causes Hydatid disease in the humans. So it is transmitted by ingestion of contaminated food with dog species. Next question, Echinococcus is the causative agent of option A, Hydatid disease. Option B, Elephantiasis. Option C, cysticercosis. Option D, intestinal teniasis. So here, what is the right answer? That is option A, Hydatid disease. So as we discussed, so Echinococcus granulosis, it causes Hydatid disease. So in the man, the larval form of Echinococcus causes unilocular Hydatid disease. So now this disease is renamed as cystic echinococcosis. It is renamed as cystic echinococcosis, that is CE. And how the infection takes place? The infection takes place by direct contact with infected dogs. And it is also transmitted by allowing the dog to feed from the same dish. So sharing of the same plate with the like a dog. And it is also transmitted by uncooked vegetables which are contaminated with infected animal feces. So here, generally this infection mostly we can see in the childhood because children are like in intimate associate with the dogs. Next question, the largest cestode found in human intestine. Option A, Diphyllobotherium latum. Option B, Taenia saginata. Option C, Taenia solium. Option D, Echinococcus granulosis. So here the right answer is option A, Diphyllobotherium latum. So Diphyllobotherium latum is the largest cestode which is present in the human intestine. So the common name is fish tapeworm, or it is also called as broad tapeworm. So what is the size of this cestode? It measures around 10 meters or more in the length. So that is the largest cestode. And how this infection takes place? By the uncooked or undercooked freshwater fish, by the consumption of uncooked or undercooked freshwater fish. Next question, what is the amount of antigen is inoculated in Casoni skin test? Option A, 0.5 ml. Option B, 0.2 ml. Option C, 0.1 ml. Option D, 0.02 ml. So here, what is the purpose of Casoni skin test? It is to diagnose the Hydatid disease, that is Echinococcus disease. So here, what is the right answer? Option B, that is 0.2 ml. So if you see what is Casoni test, it is an immediate hypersensitivity skin test. It is an immediate hypersensitivity skin test. It was introduced by scientist Casoni in the year 1911. So in this test, what we use sterile hydatid fluid. We use sterile hydatid fluid. So this hydatid fluid is taken from unilocular hydatid cyst from human cases. So that is generally removed by the operation or from the animals. And this fluid is filtered and made sterile, and 0.2 ml of this antigen is injected intradermally in one arm. And in the same time, sterile normal saline 0.2 ml is injected intradermally in other arm. So this normal saline acts as the control. In positive case, the tested site, the arm which is tested, it shows a large wheal with multiple pseudopodia within 30 minutes. So this test has low sensitivity. Sometimes it gives false positive reactions also in case of other cystode infection. Nowadays, we are not using this test. Next question. The infective form of Toxoplasma gonadei is option A, tachyzoid, option B, tissue cyst, option C, sporulated oocyst, and option D, all of the above. Here, the right answer is option D, all of the above. So here, all these forms such as tachyzoid, tissue cyst, and sporulated oocyst, they are the infective forms. So if you see what is meant by tachyzoid, it is a rapidly proliferating trophozoites in the acute infection. And how the infection takes place, how the toxoplasmosis infection takes place, it is by ingestion of undercooked meat which consists of tissue cyst and ingestion of mature oocyst through food, water, or fingers contaminated with the cat species, and even it is transmitted through the blood transfusion or organ transplantation, and even it can cross through the placenta, transplacental infection also. So transplacental transmission from infected mother to fetus, and it may cause accidental infection. So these accidental infections we can see in the persons who handle the parasites in the laboratory. Next question, detection of an antigen in amniotic fluid is used to diagnose. Option A, congenital toxoplasmosis. Option B, ocular toxoplasmosis. Option C, acquired toxoplasmosis. Option D, none of the above. Here the right answer is option A, congenital toxoplasmosis. So as I told you in the previous slide, so here toxoplasmosis, even it can cross through the placenta and it infect the fetus. So to diagnose this congenital toxoplasmosis, we check the antigen in the amniotic fluid. Next question, method of choice for demonstration of oocyst of Cryptosporidium parvum. Option A, Giemsa staining. Option B, Leishman stain. Option C, modified Gill-Nielsen staining. Option D, none of the above. Here the right answer is option C, modified Gill-Nielsen stain. So in the modified Gill-Nielsen stain, oocyst of Cryptosporidium appear bright red in the color. So this oocyst is spherical. It measures so they are measured 2 to 5 micrometer in the diameter. Next question, parasite that causes traveler's diarrhea is. Option A, Cryptosporidium parvum. Option B, Trichomonas vaginalis. Option C, Plasmodium falciparum. Option D, none of the above. So here the right answer is option A, Cryptosporidium parvum. So if you see traveler's diarrhea, it is mostly caused by the bacteria, that is Escherichia coli, and next to that one Serratia and other bacteria. But here the question is parasite which causes traveler's diarrhea is. So generally Giardia causes traveler's diarrhea, but here in these options there is no Giardia. So next to the Giardia, Cryptosporidium parvum causes traveler's diarrhea. So here the right answer is Cryptosporidium parvum. Next question, infective form of Cryptosporidium parvum. Option A, trophozoite. Option B, tissue cyst. Option C, oocyst. Option D, precyst. Here the right answer is option C, oocyst. So how humans get the infection? They get the infection by ingestion of food and water which is contaminated with the feces which consists of the oocyst. So here oocyst, it is of two types, thick-walled oocyst and thin-walled oocyst. And if you see thin-walled oocyst, it causes auto-infection, and thick-walled oocyst causes infection in the new host. Next question, Plasmodium knowlesi causes. Option A, malignant tertian malaria. Option B, benign tertian malaria. Option C, quotidian malaria. Option D, none of the above. Here the right answer is option C, quotidian malaria. So what is quotidian malaria? So here we can see the fever spike for every 24 hours. For every 24 hours, the patient will get the fever. So Plasmodium knowlesi causes quotidian malaria. Next question, common fluorescent stains used for detection of Cryptosporidium. Option A, oromine phenol. Option B, acridine orange. Option C, both A and B, that is oromine phenol and acridine orange. And option D, none of the above. Here the right answer is option C, that is both A and B. Oromine phenol and acridine orange stains, they are used for detection of Cryptosporidium. Next question, Plasmodium malariae causes. Option A, quartan malaria. Option B, benign tertian malaria. Option C, quotidian malaria. Option D, malignant tertian malaria. Here the right answer is option A, quartan malaria. So quartan malaria, so Plasmodium malariae causes quartan malaria where fever occurs an interval of 3 to 4 days. And in case of Plasmodium vivax and Plasmodium falciparum, so generally the shy giants mature within 48 hours. So that's what fever occurs after 48 hours. But in case of Plasmodium malaria, that fever occurs after 3 to 4 days. Next question, dark pigmented granules are seen in which form of Plasmodium vivax? Option A, ring form. Option B, trophozoite. Option C, gametocyte. Option D, none of the above. Here the right answer is option B, trophozoites. Dark pigmented granules are seen in the trophozoite form of Plasmodium vivax. Next question. A mature shy giant contains usually dash merozoites. Option A, 2 to 4. Option B, 5 to 6. Option C, 100 to 200. Option D, 12 to 14. Here the right answer is option D, 12 to 14. A mature shy giant consists of 12 to 14 merozoites. Next question, most sensitive method for malarial detection. Option A, Kawamoto technique. Option B, QBC. Option C, Leishman staining. Option D, none of the above. Here the right answer is option B, QBC method. So what is QBC method? That is quantitative Buffy coat test. So quantitative Buffy coat test, it is the new method for identification of malarial parasite in the blood. So in this test, what we do? We collect the blood in the QBC tube coated and the QBC tube, it is coated with the fluorescent dyes. And after that, it is centrifuged. After centrifugation, the tube is examined under fluorescent microscope. So acridine orange stained, the malarial parasites appear brilliant green in the color. And it is more sensitive method than the thick blood smear. And it can detect 3 to 4 parasites are also present in the microliter of the blood sample. But the disadvantage is, so here the species cannot be identified, and it is costly also. And if you see Kawamoto technique, that is fluorescent microscopy. So that is a fluorescent staining method for a demonstration of malarial parasite. In this method, the blood smear is prepared on a slide and it is stained with the acridine orange, and the stained slide is examined with a fluorescent microscope. Nuclear DNA of malarial parasite is appear green in the color and cytoplasmic RNA appears red in the color. And if you see Leishman staining, so Leishman staining, it is done for thick smear examination. Next question, recent technique to detect circulating malarial parasite in blood. Option A, ELISA inhibition test. Option B, Kawamoto technique. Option C, QBC method. Option D, none of the above. Here the right answer is option A, ELISA inhibition test. Next question, main difference between the life cycle of Plasmodium vivax and Plasmodium falciparum. What is the main difference between the Plasmodium vivax life cycle and Plasmodium falciparum life cycle? Option A, hypnozoites. Option B, trophozoites. Option C, merozoites. Option D, none of the above. Here the right answer is option A, hypnozoites. So what are the hypnozoites? Hypnozoites, they are the resting stages. They are They are formed in the exoerythrocytic schizogony. And if you see Plasmodium vivax, it has exoerythrocytic schizogony. And if you see Plasmodium falciparum, it doesn't have exoerythrocytic schizogony, which means in case of Plasmodium falciparum, hypnozoites are absent. And in case of Plasmodium vivax, hypnozoites are present. So these hypnozoites are responsible for relapses in malaria. So that's what we can see, the relapses in case of Plasmodium vivax infection, and there is no relapses in the fever in case of Plasmodium falciparum infection. That is the main difference. Next question, presence of erythrocytic shy giant in peripheral blood smear shows. Option A, acute infection. Option B, relapse. Option C, severe infection. Option D, none of the above. Here the right answer is option C, severe infection. So presence of erythrocytic agent in peripheral blood smears shows that severe infection. Thank you.